This lecture is going to be about vectors and expression systems for protein expression. And we're going through, as always, through Google Images and through things that we find on the internet, because that's really how we're learning now. And all the labs and all the companies are posting to all these things. So, you know, that's how we really do our learning now. So we're going to start off just with a very much of a backbone sort of expression system. These are called pet vectors. And so with the pet vectors, what we have is, let's bring one up here. Okay, with the pet vector, we're going to have, it is going to be an E. coli. And these were actually invented in the late 60s, 1980s. And, but they're still used a lot today. It's kind of the backbone of expression in bacteria. And so what we have is it's just a standard plasmid. And so if you look at this, we have an ampicillin resistance gene, and that is, what would we call an ampicillin resistance gene? What is the category for that as far as a plasmid is concerned? What's the use of that?
Selectable marker.
A selectable marker, very good. And then the ORI, what is that there for? Origin of replication. So if you have an ORI site, then you're going to be replicated. And then the LAC-I, or the LAC-Z gene in there. Well, actually, that's going to be for, sorry, that's not the LAC-G, it's not the multi-cloning site. In this case, the F1 ORI is there just because this makes this into a phage mid. So the F1 ORI is there just so that it can serve as, to create single-stranded DNA if you wanted to sequence this, for example, or use it as a probe. And so that's what that is there for. It's the F1 phage. And then what we have here is the, next up is we have a promoter for T7. So it's the promoter that responds to T7 RNA polymerase. And this is the center technology for pet vectors. So T7 RNA polymerase, would you find that in E. coli? Just normal T7 RNA polymerase. Where does T7 RNA polymerase come from?
A virus.
From a virus, exactly, from bacteriophage T7. So it's a very powerful polymerase. And the promoter is, of course, very, very strong because it's a virus. But what they did is they have special cells, special bacterial cell lines that express T7 RNA polymerase. So this vector does nothing if you put it in a normal E. coli, but if you put it into a T7 RNA polymerase containing cell line, bacterial cell line, then you will get a lot of expression. Now, was it good to have your foreign protein expressed all the time? Would there be any detriments to having your gene of interest protein being expressed all the time? Because if you put this in one of these cells that has T7 RNA polymerase, it's going to go from the get-go, it's going to express. Can you think of any problems you might have with expression, constitutive expression, as they say?
Use up all the substrate.
You could use up all the substrate, that's true. And what sort of proteins are medical scientists usually interested in? Dangerous proteins. Proteins that are detrimental to life. So we're interested in proteins that turn things on, proteins which might be toxic, and everything else. That's generally what happens. So we're going to have, instead of a constitutive promoter, we're going to have an inducible promoter. And I'm not sure if we can look over here and see if we have any other ones listed here. I don't think we do. But basically, we have, let's see. Yeah, here we go. Okay. So if we look at this one, what we have is we have a different promoter which is driving the production of T7 RNA polymerase. And then that RNA polymerase will land on this promoter, the T7 promoter, and then we get our protein. But this particular promoter, this is PTAC promoter, and there are other inducible promoters as well. But this one is completely dependent upon IPTG because it is a combination of two promoters but one of those promoters is the tryptophan inducible promoter and the lac inducible promoter. This one is it's a very powerful promoter. It is induced by IPTG, which is a substitute for lactose. You could add lactose and you would turn this promoter on as well. But IPTG is what really turns this thing on. And so if you add IPTG, then you get T7 RNA polymerase being produced. Then you get your protein of interest being produced. And that's how pet vectors work. Okay, let's go on then to yeast vectors. Any questions before we leave this subject? Okay, with the yeast vectors, we're going to look at 3 different types. We're going to start off with a 2 micrometer vector. So it's hard to write micro in your Google search. Oops, and now we have 2 micrometers. Oh, we have our calipers. So 2 micrometer vector yeast. And here we have it right here. So we have a 2 micrometer circle, and you'll see from this that you have, let's go to the original page on this. This is a nice page. So you have, this is actually a plasmid. And so this page is titled Plasmids in Eukaryotic Microbes. Now plasmids we always think of as being involved in bacteria, right? And so we always think of that, okay, plasmids are in bacteria, but there is a plasmid which is in a eukaryote, and that's in yeast, and that's a 2 micrometer plasmid. It acts just like a regular plasmid. It has the origin of assembly, the ORI site, but in yeast language we call that ARS, autonomous replicating sequence. So that's different. And then we have these other proteins which are responsible, the rep proteins which are responsible for replication. But what is really important here, so it's responsible for replication, and then this thing replicates just like a plasmid in eukaryotes. Now, plasmids generally replicate by bidirectional replication. So you start off with the replication bubble. So you have the whole circle. And actually, we can go over here. Just a minute. Plasmid replication. Bidirectional. Let's look at that. So normally, we have something that looks like this. So we have the origin of replication, and then we have these replicating forks here, right? There's the origin of replication. And we just keep on going and going until we finally finish after this. So there are many copies of this eukaryotic plasmid in the yeast. But yeast only multiplies once in a while. So how do you get more than one copy of this 2 micrometer plasmid in yeast? That's an interesting question, because you'd only replicate once and then that would be the end of it. So the way this works is we have these regions right here, and those regions will, and then there's this region called flip right there. And what this does is that you'll notice that what the replication begins, let's say right here at this A point. And then you have one fork going this way, another fork going this way. Well, one fork, of course, traverses this portion and is going in this direction, so it's going in that direction, right? It's going to the left. The other fork is going to the right. Then this flip recombinase flips this thing over. It actually does a 180. So the F is up here. Now the F's down here. The D is down here. And now the D is up here. So it just spins it around and then reconnects it. Well, the replication fork was going this direction to the left on F, right? Now it's going to the left on F here. That means it's going here. It was going counterclockwise in the circle. Now it's going clockwise in the circle, in the same direction as the other fork. So the forks will not go, one goes clockwise, one goes counterclockwise, they meet at the other end. Now they're both going in the same direction. They go round and round and round and chase each other. And so we can have many, many, many copies of this plasmid. So that's what the flip is there. So that's a 2 micrometer circle, and you can use these in yeast to be able to produce lots and lots of protein, and you can just put a gene into this 2 micrometer plasmid and transform yeast and get your protein. And if you don't like it, well, you just go back and make more. You can do mutagenesis studies, everything. We're using it in our lab right now in Saccharomyces cerevisiae. And it's a very nice little system. And the other thing is most of these 2 micrometer plasmids with yeast vectors, they also work in bacteria. So not only do they have an origin of replication, the ARS for yeast, they also usually have one for bacteria as well. And they've got like an ampicillin resistance gene and these sorts of things. So they'd be called what we call a shuttle vector. You can make them in bacteria, do all your genetic engineering, and throw them over to yeast, and they'll grow and grow and grow and produce lots of protein. Well, the other question then is, well, what if you can't actually use these 2 micrometer plasmids in yeast because they're unstable? Because like all plasmids, they can be lost and other things like that. So what are we going to do about that? And what you can do is you can use a YIP vector, and that would be a yeast integration plasmid, a yeast integration plasmid, and YIP vectors work this way. Or YIP vectors and YEP vectors, they can both work this way. And so I just have the YEP vector up here. But the way this would work, YEP vectors are also episomal, so these things can as well act like a 2 micrometer plasmid. But for both of these, YEP and YEP vectors, they would contain a gene which is also found in the yeast chromosome. A gene which is also found in the yeast chromosome. So if you clone your gene into this thing, then you can go ahead and add it to the yeast. And it will do something called homologous recombination with the leucine gene in yeast. Except here's the trick. The yeast strain you used has a defective leucine synthesis gene. Defective leucine synthesis gene. So it will only grow on minimal medium supplemented with leucine. That's the only medium it will grow on. And, but when we add this YIP or YEP vector, which has leucine 2, it's a wild type leucine gene. So if you do homologous recombination, you will wind up getting a functional leucine gene out of the deal. One of these will be non-functional, one of these will be functional. One of them will carry the mutation found in this one. The other one will be completely functional, right? Because there's only one mutation in there, right? So one of them gets the mutation, the other one is functional. So the point is you now have a functional leucine synthesis gene. You can grow this on total minimal medium. In the absence of leucine, it should work. So what would we call this leucine gene? You mentioned it earlier at the beginning of the lecture. You gave it a name. The leucine gene would be analogous to some other gene. It's analogous to ampicillin resistance, right? So what would we call this? It's a selectable marker, exactly. So why can't we use ampicillin resistance in yeast? What's the difference between yeast and bacteria?
Yeast is a fungus.
Yeah, yeast is a fungus, so it's a eukaryote. So ampicillin is not going to work against it. Ampicillin is an antibiotic. Most antibiotics work against bacteria. Yeah. That's why when you have like a toenail fungus infection, it's really difficult to get rid of because you can't use antibiotics. They're yeast. They're eukaryotes just like you. And so you can't use this. So we have to resort to other things. Now, kanamycin can be used, and we'll see that in a later discussion when we talk about antibiotics against plants. But and it's also used against humans. But here we're using this system. Okay, so that's how that works. And those are YIP vectors, but in this case it's a YEP vector, which is also a plasmid. But it serves the purpose. You see how it works. Okay, finally we're going to go to